Hello everyone, I am Tejashwini from 2nd MSc Microbiology. I will be giving seminar talk on the topic Pseudomonas fluorescens as biopesticides under the guidance of Dr. H. Pushpa ma'am. Let's start with general introduction. Biopesticide, by the word it means biological pesticides which are naturally occurring substances, includes several types of pest management intervention. This is through predatory or parasitism relationship by non-toxic mechanism. Biopesticides are the one which are used by manipulation of microorganisms such as bacteria, fungi, microalgae, nematodes, etc. Since they do not affect the plant and in fact they reduce soil pollution and also erosion, it is very much required for sustainable agriculture. They are also less toxic for the user and non-target organisms. Biopesticides include several types such as microbial pesticides, whereas bacteria, fungi or protozoa acts as the active ingredient. Plant incorporated protectants. These are the genetically modified plants, whereas the genes that help the plant bodies to produce some substances to fight against pests. Biochemical pesticides. This includes substances such as insect sex pheromones. Biotic agents. This includes predators and parasitoids which are active against pests. Microbial biopesticides. As mentioned earlier, these biopesticides are composed of microscopic living organisms such as virus, bacteria, fungi, protozoa or nematodes. Sometimes the toxins produced by these organisms are also the active ingredient. They are relatively specific for the target pest and essentially non-toxic for the user or the plants and non-target animals. They constitute the largest group of broad-spectrum organisms which are pest-specific. Many microbes are used as biopesticides or biocontrol agents, particularly soil-borne diseases of plants are controlled by the usage of biocontrol agents. Many fungi and bacteria are used as biocontrol agents. To state among them, Trichoderma, Gliocladium, Ampelomyces are few fungal bioagents, whereas Agrobacterium radiobacter, Bacillus subtilis, Bacillus thuringiensis, and Pseudomonas fluorescens are few important bacterial biocontrol agents. Pseudomonas fluorescens as biopesticides. Pseudomonas fluorescens in particular have gained importance as a component of integrated pest management for sustainable agriculture. Systematic classification: domain bacteria, phylum Proteobacteria, class Gamma Proteobacteria, order Pseudomonadae, family Pseudomonadaceae, genus Pseudomonas, and species we are going to study is Pseudomonas fluorescens. To state the general characteristics, Pseudomonas fluorescens is a rod-shaped gram-negative bacteria which are obligate aerobes. There are certain strains which are capable of using nitrate instead of oxygen as final electron acceptor during cellular respiration. They possess bipolar flagella and they are motile. They show an extremely versatile metabolism and can be found in the soil and in water. It is an non-saccharolytic bacteria which shows positive for oxidase test. The optimal temperature for the growth is 25 to 30. They produce heat-stable lipases and proteases enzyme which causes milk spoilage, casein breakdown and ropiness due to the protein coagulation. The specific name fluorescence refers to the microbial secretion of a soluble fluorescent pigment in this organism which is known as pyoverdin. It is a type of siderophore. It is this siderophore which make them an active biopesticide. Under the condition of low iron availability, it secretes soluble greenish fluorescent pigment called fluorescein. Pseudomonas fluorescens belong to plant growth promoting rhizobacteria (PGPR), an important group of bacteria that play a major role in the plant growth promotion, induce systemic resistance, biological control of plant pathogen, etc. These bacteria, since they reside around the roots of the plants or the crops, they aid the plants in several ways such as destroying certain toxins and pollutants including styrene, polycyclic aromatic hydrocarbons and TNTs. It has been theorized that Pseudomonas fluorescens could be a good alternative to synthetic pesticide because of its toxicity to larvae and pupa of Vector mosquitoes, which have been two concerns in the agricultural business. There are certain characteristics possessed by Pseudomonas fluorescens which made them as prominent biopesticide, and due to these traits they are well suited for the role of biopesticide and as well as growth promoting agent. They grow rapidly in vitro and to be produced in mass. They rapidly utilize seed and root exudates, and they also colonize and multiply in the rhizosphere as well as in the spermosphere by adhering to the soil particles and to the interior of the plant. They produce a wide spectrum of bioactive metabolites, mainly the secondary metabolites. The production of hydrolytic enzymes also aids as the biopesticide. They also compete to the environmental stresses, and they possess PGPR traits such as nitrogen fixation, phosphate solubilization, ion chelation and phytochrome production, and mainly they are also inexpensive to be used as biopesticide. Now let's study the mode of action of Pseudomonas fluorescens which made them to emerge as biopesticide. The mechanism of action of these organisms is indirect, which involves the production of antibiotics, production of siderophore, induced systemic resistance, competition towards other microorganisms, hydrogen cyanide production, and plant growth antibiotic production. Antibiotic production. Pseudomonas fluorescens produce many secondary metabolites which acts as antibiotics against plant pathogens or plant pests. The fluorescents produce antifungal compounds which are fungistatic and which inhibit the germination of fungal spores, and they also lyse fungal mycelia. Phenazine 1 carboxylic acid (PCA), 2,4 diacetyl fluoroglucinol (DAPG), phyocyanin, phylonitrin, oomycin A are few of the antibiotics which are produced by fluorescent pseudomonads. These antibiotics have effective causes on the pathogens. Phenazine 1 carboxylic acid is a nitrogen containing secondary metabolite which possesses redox activity and can suppress the growth of Fusarium oxysporum, and it is used to control the take-all disease of wheat which is caused by Graminis. DAPG, that is 2,4 diacetyl fluoroglucinol, which is one of the effective antibiotic, causes membrane damage to Pythium species, and it is particularly inhibitory to the zoospores of oomycetes. Phyocyanin, it is one of the toxin produced by Pseudomonas genus which is blue in color, and it has the ability to kill the microbes by competing against them by generating reactive oxygen species. Phyoleurin is most notable for its toxicity production of siderophore. It is one of the important mode of action which made Pseudomonas fluorescens as a prominent biopesticide. Siderophores are secondary metabolites produced by different organisms in order to scavenge iron from their surrounding environment, making this essential element available to the cell or the host plant. Presenting high affinity for ferric iron, siderophores are secreted out to form soluble ferric complexes that can be taken up by the organisms. Siderophores present complex chemistry that allows them to form the strongest ion chelating complexes. Siderophores participate to the mobilization of iron and other elements and are involved in virulence process also. There is a strong relation between siderophores and oxidative stress tolerance which has to be highlighted here. These siderophores bind to Fe3 plus ions and transport them into the cells. Fe3 plus is a biologically important molecule as it is an important constituent of cytochrome, heme and non-heme protein, and a cofactor for various enzymes also. The PGPR under iron deficiency synthesizes this siderophore to sequester iron. Most common forms of siderophores are catechols and hydroxymates. These siderophores produced by Pseudomonas fluorescens have ability to stimulate plant growth either directly by increasing the availability of iron in the soil or indirectly by inhibiting the other competitive plant pathogens which have less efficient iron uptake system. Pseudomonas fluorescens produce many types of siderophores. Among them, pyoverdin is important. Ferribactin, ferrichrome, ferrioxamine are other siderophores which are also produced by fluorescent species. The potent siderophore pyoverdin shows a higher affinity for iron and can inhibit the growth of both fungi and bacteria which have less potent siderophores in iron depleted media. The siderophores produced by these organisms can control the potato decay disease which is caused by Erwinia species and Takol disease caused by Graminis. Induced systemic resistance. It is a resistant mechanism which gets activated in the infected plant. The mode of action depends either by killing or inhibiting the invading pathogen and conferring a physical and chemical barrier of the host plant which gives long protection against broad spectrum of microorganisms. The PR proteins such as chitinase, glucanase are used as ISR marker which is only produced by infected plants. The signaling compounds which are responsible for the induction of these PR proteins are salicylic acid, ethylene or jasmonic acid. All this shows effective antagonism against necrotrophic pathogens and insect herbivores. The passive or the wound responses of this systemic resistance includes production of waxes, occutin, phenolic compounds, and quinones. Once after getting the infection, there is an increased accumulation of phytoalexins or free radicals or calcium or peptides. We know these phytoalexins acts as antimicrobial and antioxidative substances which accumulate rapidly at the areas of infection in the infected plants. Different strains of Pseudomonas fluorescens shows this induced systemic resistance against many target pathogens such as Oxysporum, Xanthomonas campestris, tobacco necrosis virus, etc. Hydrogen cyanide production. HCN is also a representative class of volatile inhibitors. Pseudomonas fluorescens produce hydrogen cyanide which can inhibit the growth of many phytopathogens. There are several studies which shows this hydrogen cyanide is used in chelation of metal ions as well as it is indirectly involved in making availability of phosphate to the host plant. The cyanogenic strain of Pseudomonas fluorescens is reported to have a potential role in biocontrol and in the enhancement of stem and root length. They also have a germination rate of rye, wild barley and wheat. Competition. Pseudomonas fluorescens by their establishment in the rhizosphere zone gives competition or antagonistic nature to the other fungal and bacterial species which acts as pest for the host plant. They give competition for favored sites on the roots and also in the rhizosphere. They are competitive for the other nutrients uptake also. Plant growth promotion, antibiotic production. Our Pseudomonas fluorescens promotes growth by the production of phytohormones such as auxins and gibberellins. We know these auxins are important phytohormones which promotes the growth and developmental events and they also improve plant tolerance to the abiotic stress. Gibberellins are the hormones which synthesize and produce hydrolysis and amylases which helps in the germination of seeds. Isolation and multiplication. The isolation of Pseudomonas fluorescens is by collecting the soil from the rhizosphere, whereas the serial dilution technique is carried out. King's bee medium is well suited for the culturing of this organism. This culture can be mass multiplied on king's bee broth for 48 hours at 25 or 30 degrees Celsius. The pH is adjusted to 7. Carboxymethyl cellulose is added in the substrate at 10 gram per kg. The autoclaving is done for 2 consecutive days for about 30 minutes. The product is dried under the shade to bring the moisture content to the less than 20%. The pack or the formulated product is in white colored polythene bags and it is sealed and kept under room temperature. The fresh products can contain up to 3 into 10 power 3 colony forming units per gram of Pseudomonas fluorescens. There are several commercially available Pseudomonas fluorescens biopesticides in the market. Few of them are Monas, Pseudo, BioShield, Pseudocon, Biomass, which are the trade names of biopesticides produced using Pseudomonas fluorescens. Let's conclude. There are several environmental and consumer concerns which have been focused on the development of biological agents as an alternative and eco-friendly strategy for the production and the protection of agricultural business and horticultural crops which are against phylopathogens. Pseudomonas fluorescens is one of the such proven biological control agent. There are many successful reports which shows around the world have described different Pseudomonas strains are able to control a number of fungal and bacterial diseases in cereals. Peat soil. It is found to be the best substrate for the colonization of Pseudomonas fluorescens. The polysaccharide enhanced the addition of this organism which promoted plant growth through increased antibiotic activity. There are many reviews which contributes to the future research programs that can aim to the promotion of this Pseudomonas fluorescens as a potential biopesticides for augmentative biological control of many diseases of agricultural and horticultural importance. These are the reference for this particular topic. Thank you.